we are going to finish up the autonomic nervous system by touching on the higher centers of the brain that have fairly direct controls over regulation of the autonomic nervous system. And then we're going to look at autonomic reflexes and compare them to the somatic ones we've already talked about. And then we're going to talk about special senses. So when we look at upper level controls and how the central nervous system influences the autonomic, we see that even though the autonomic nervous system is by definition not under voluntary control, the central nervous system still is in charge of it. We're just not consciously aware of it, and we can't consciously control it. The major portion of the central nervous system that directs autonomic function is the hypothalamus. So it's the major integrative center for the autonomic nervous system, and as we'll talk about next week, it's also the major controller of the endocrine system. So really when we look at homeostasis and regulating quite a lot of body functions, the hypothalamus is a major, major player. Hypothalamus, I say this. The cerebral cortex can also have effects. Again, even though we can't consciously control this stuff and we aren't really even consciously aware of it, our emotions have a role in influencing autonomic effect. And so via our limbic system, because of our emotions, or sometimes by our hypothalamus, we can exert effects. And basically what that boils down to is most of the time it means that if we're having strong emotions, we're going to have physiological responses to them because of this connection. So if you are really scared or really mad, we're going to be stimulating the sympathetic nervous system because those are forms of stress. And when you're feeling really happy or calm or something like that, then you're probably going to have higher parasympathetic tone because your emotional state says everything is safe and fine. So that's sort of the why and how of that interaction. So this is showing you a little bit of how that connection works. So here's your hypothalamus right here. And there's going to be some connections to the limbic system and the cerebral cortex. There's going to be connections to nuclei in the brainstem controlling, and the spinal cord controlling various autonomic functions. And we don't need to know the details about that right now. So again, the limbic system connection, it's about having appropriate autonomic responses to emotional stresses. And the thing about this, and again, this is where there's a lot of overlap between the nervous system and the endocrine system. Not only will we get stress-related increases in sympathetic tone, so increasing sympathetic nervous system tone, but we'll also get sympathetic hormone release by the adrenal medulla, so that's epinephrine and norepinephrine. And we'll potentially be getting stress hormone release, which, as we'll talk about later, is also mostly from the adrenal gland, but it's from the cortex. So we'll talk about that on Monday when we get to the endocrine system. This is just a little very brief thing about how we can learn to sometimes have some conscious control over some of our homeostatic mechanisms. So biofeedback is where you are measuring something like heartbeat, heart rate, which is what this is doing, or sweating or something like that. But heart rate's a very common one. And you're seeing that displayed, and being able to watch it display in real time, you can learn to alter it. So people can learn to slow down their heart rate. That's usually the purpose of this. And we call that biofeedback, this idea that you can kind of learn to influence consciously these unconscious processes by becoming aware of them. So just want to touch on that as another example of higher level control, but it does take training. All right, so visceral or autonomic reflexes, they are both the same and different from somatic. So they have the same 5 components. All reflexes have the same components. There's a sensory receptor. a sensory neuron, an integration center, a motor neuron, and an effector, always. Now there's some differences. So we're going to be using smooth muscle, heart muscle, and glands as our effectors. The autonomic output always uses those two motor neurons, so the pre and post ganglionic motor neurons in the pathway, whereas the somatic only uses the one motor neuron. And then the sensory input, the receptors and sensory neurons, are going to be for visceral input. So we're getting input from enteroceptors rather than proprioceptors or exteroceptors, which are the ones that would be mediating the somatic reflexes. So now we're going to see reflexes in response to chemical changes within the body, to stretch of hollow organs or something else like that, to irritation and inflammation of viscera, which is another way to talk about visceral organs, and other things like that. Whereas with somatic reflexes, we're looking at things like responses to pain on the skin and, you know, stretching or other things affecting muscles and tendons and joints. So this is our breakdown of comparing those two things. So the autonomic up here, two motor neurons for our output with a ganglion and synapse in between. And instead of having a proper synapse like a neuromuscular junction, autonomic nerves tend to end in these varicosities and have a more diffuse release of neurotransmitter as they control smooth muscles. That part's not super important. And then here's our single motor neuron and neuromuscular junction to control skeletal muscle. So this is one from another textbook showing the 5 steps, and it's specifically for an autonomic pathway, but it's showing you the same stuff. And it's also showing you how our motor neuron cell body would be in the lateral horn of the spinal cord, because that's where autonomic motor neurons originate, and that we would be synapsing in an autonomic ganglion, and then the postganglionic goes to the visceral effector. So let's look at an example. We're going to use one that we can kind of practice at home if we really were curious about it. So it's called the pupillary light reflex. And basically, when bright light hits the back of your eye, the retina, the part that actually detects light, the muscles in your iris that make your pupil contract, and so your pupil shrinks. And this is because that muscle is under parasympathetic control, and its purpose is to decrease the amount of light entering your eye, because when you are exposed to really bright light, you need less light in your eye to be able to see clearly. Otherwise, you get dazzled. It's too bright. Now, the effect is bilateral. And what that means is, regardless of which eye is hit with the bright light, both eyes will constrict. Both pupils will shrink. So the reason why this happens is because of the way that the reflex is set up. So this is kind of an overview of the reflex, and then we're going to look at one that shows us why both eyes are going to do it. So our sensory input here is going to be light, and our sensory receptors are going to be the nervous tissue of the retina. And we'll talk a little bit more in detail about that when we get to vision. We're not going to worry about it too much right now. So the sensory neurons are going to go into the brain, and then don't worry about this. This is in dim light. We don't care right now. When there's bright light, there's a midbrain nucleus. So this is going to be our integration center. Okay, so sensory receptor integration center. And then our 2 motor neuron output goes back to the smooth muscle in the iris, which constricts and causes pupillary constriction or shrinkage, okay? So 1, 2, 3, 4, 5 reflex steps, because every reflex has the same 5 steps. Did I put it in there? I did not. Okay, sorry. I thought there was a chart in there that there wasn't. Anyway, the reason why this happens bilaterally is because both eyeballs send information to both sides of the brain. So regardless of which eyeball gets the information, the response is integrated and carried out bilaterally, okay? Hey, stop it. All right, so that's just one example. Other examples, pretty much any negative feedback loop. We've talked about a little bit at the beginning of the class with homeostasis. Those are autonomic reflexes. So when we talk about how, you know, like your body adjusts for something like blood pressure or thermoregulation, those are basically just autonomic reflexes being carried out, and we're talking about them in a different way. Another thing that is kind of weird and interesting about the autonomic nervous system and visceral sensations. So the input into the brain from visceral sensory receptors is that even though we're not usually aware of it, they are still heading back into the brain and informing the brain of visceral input. And so when you have strong enough visceral sensations, and honestly, it's usually pain, so strong enough pain, it can enter conscious awareness. So sometimes we can feel visceral pain, and we don't always feel it in an accurate way. So when we feel somatic pain, we know where it's coming from because we have that very accurate map of where all of our sensory receptors are coming from as they enter the brain, somatotopy. We don't have that for visceral receptors. So all we get is this input of pain. And because the pain nerves, because those axons are traveling into the spinal cord and up to the brain when in the same pathways as somatic, often we get this cross stimulation of the somatic nerves, and we feel like the pain is somatic when in fact it's visceral. This is so consistent that we call it referred pain or referred visceral pain, and there are maps of the common areas on your skin that we can feel pain that is actually internal. So for example, and these aren't exactly the same for everybody, okay, but they're fairly consistent. So for example, this whole area of the left side of your chest and your left arm, this is where we're commonly feeling referred pain when it's actually coming from your heart. And that's why one of the classic signs of heart attack is left-sided chest pain, left arm pain. You can see there's actually even a little spot right there in the middle of the back. Now, especially for women, this actually goes all the way up to here, and you can have jaw pain and neck pain as well. So this is not like 100% for everybody, but this is sort of a general average map. You can see, for example, that the kidneys have a very broad area of referred pain. That's basically the whole pelvis and parts of the legs. And so this is something that doctors can use because, you know, if your arm hurts, but you didn't do anything to your arm, and maybe you have some other things like shortness of breath, or maybe you feel nauseated or something, then they can be like, well, it's because you're having a heart attack, not because you injured your arm, right? So that's both the usefulness of it and also why it's very weird that way. So referred pain, because we don't have as accurate of a map of visceral sensations as they are entered and processed by our brain. So that was kind of the rest of autonomic. And these were a couple of interesting links if you're curious, mostly about perception. And so now we're going to go over to special senses and talk about the rest of the sensory input to the body. So in our free textbook, the majority of chapter 14 is special senses as well as general senses and like motor output. And I like to just tackle the special senses on their own, so that's why we're doing it separately and last. So we're going to start with the chemical senses because they're the easiest. So when we talk about the special senses, we're talking about taste, we're talking about smell, hearing, balance isn't quite accurate, but it has a lot of different names, hearing, equilibrium, and then of course we're talking about vision or sight. These two are chemical senses, so they're using specialized chemoreceptors. And we'll talk about what the other guys are after we get through that. So we have two chemical senses that use specialized chemoreceptors, but they do it in different ways for different purposes. Now, both of them require that the chemicals that they have receptors for be dissolved in solution. So even though when we smell things, we're smelling chemicals in the air, those chemicals actually have to dissolve in the nasal secretions, so mucus and a little bit of water and stuff, in order to actually bind to the receptors that can detect them. Now, smell and taste respond to different classes of chemicals, and that's because they exist for different purposes. So taste, or gustation, which is the fancy term for it, is for identifying things we put in our mouths. So mostly it's for the identification of food and drink, although I would argue that when we evolved this, mostly we drank water. But mostly for food, and it's to make sure that we are eating things that are nutritious and high in calories, because starvation was the most common danger, food-related danger in the past, and also to make sure that things we were eating weren't likely to kill us, that they weren't poisonous or toxic in some way. So our taste receptors are located in little structures called taste buds, and those are on physical structures on the tongue called papillae. So a papilla is just a bump. Now we do have taste buds in other parts of our oral cavity, including the insides of our cheeks and our palate, and then there are some at the back of our throat in our pharynx and epiglottis, but the majority of them are on our tongue. There's actually 4 different kinds of papillae. They are helpful for adding texture to our tongue, which should help us chew and swallow food. And they're just named for their shapes, but they all kind of just look like this, to be honest. So the circumvallates, they look like little hills inside of valleys. There's a little circly guy. And the foliates look like leaves, basically. And the filiforms kind of look like whiskers, and the fungiforms look like mushrooms, okay? The filiforms don't have taste buds on them, but the other guys do. And they're located in different areas. So the majority of the surface of the tongue has the fungiforms. The filiforms are on the edges. The foliates are, for some reason, on the sides here. And the circumvallates are those big guys on the back of your tongue. The taste buds are these little guys that kind of look like little onions. So we have a bunch of epithelial cells here, and they have these little microvilli, and then the neurons that synapse with them are down here. So it's about 50 to 100 epithelial cells, and most of them are the taste cells. So there's specialized cells that are not nervous tissue, whose job is to act as the sensory input for taste. So their long name is gustatory epithelial cells. Taste cells is what we call them because that's a mouthful. And they have these microvilli on the very top, which actually have the receptors for the different chemicals that we can taste. Don't worry about this, we're not gonna really talk about it. There are also these basal epithelial cells that act as stem cells to replace these cells. Off the top of my head, I want to say they get replaced every 7 days or so, but I'm probably wrong. Maybe, I don't know, doesn't matter. Anyway, and then at the base, we have the dendrite, so the receiving end of the sensory neurons. And that's how we end up carrying this information back to the brain. So here's another one of the taste buds. These are different papillae over here, showing you where the taste buds are located on the different types of papillae. You don't need to know that detail, I just like pictures. So here's a different taste bud. So the taste cells are these little guys, and then their microvilli are up here. There's just a little entrance into the taste bud called the taste pore. All the receptors are going to be here. And then the synapses with the sensory neurons are going to be down here because their dendrites are coming up like this. And then the neurons go off that way. So some things about taste, the density of taste buds does vary. So people that have a very high density of taste buds are so-called super tasters. So they're very sensitive to taste differences, and a lot of foods can taste very bitter or too strong because literally they have more taste buds, and so everything seems a lot stronger. Some people are on the other end of the spectrum and have a fairly low density of taste buds, and we call them non-tasters. And so a lot of foods taste bland. They need very strong tasting food to really taste stuff, but probably most people are somewhere in between there, and you know, that's totally okay too. Here's some interesting articles about that, because there's a couple of chemicals that we use to measure that, but it doesn't really have a lot of implications beyond whether you're likely to be picky about your food or not. So give those a read if you're curious. Otherwise, don't worry about it. So you probably have some idea of how this works, but the types of things we can taste are called the modalities, taste modalities. And so we can taste sweet stuff, which is mostly our ability to taste mono and disaccharides or simple sugars. Although some alcohols and some amino acids, the chemical saccharin and other things that are qualified as artificial sweeteners will also trigger the same receptors. And so all of the artificial sweeteners that we use work because they hit these receptors. Sour is triggered by acid. And of course, usually that's in something like citrus fruit. Salty is triggered by things like sodium chloride. And of course, that's very important because we need enough salt intake. Bitter is triggered by a class of organic molecules called alkaloids, as well as things like aspirin. And the reason why we can taste bitter is because most things are toxic and we shouldn't eat them. But of course, sometimes we do that anyways because things like coffee are very bitter. And then the fifth one is called umami, and it's a savory type flavor, if you haven't heard of it. And so the things that we have, basically what triggers the receptors are a couple of different amino acids, most notably glutamate and aspartate. And so this is sort of our ability to know when something's meaty or high in protein. That's what it typically correlates with. Now, it turns out that there's actually 7 modalities, but we keep teaching 5 because the last 2 have only been detected in the last few years, and they're still working everything out, and so they haven't made it into the general textbooks. But we can technically taste lipids. So we have receptors for triglycerides and fatty acids, and we also have receptors for larger carbohydrates. So they're called starch breakdown products. So we can't taste starch, you know, like what you would find in a potato, but we can taste the chains of glucoses before they get fully broken down into simple sugars. So those 2 have both been studied fairly recently, and at some point we'll have 7 modalities in the textbooks. I did put them in. I thought I pulled them out. Okay, cool. Oh, okay. Sorry, I meant to delete that. Didn't delete it. Anyway, last thing about taste. All your taste buds pretty much detect all the things. So there's no, there aren't areas of your tongue that detect different things better. That's a weird myth I don't know how it got perpetuated because you think it'd be fairly easy to disprove, but anyway, that's not the way it works. Everything tastes everything. And then this is just a reminder, we talked about cranial nerves before, so 3 different cranial nerves carry taste sensation from different areas, and it all ends up in your brainstem and then going up to your cerebral cortex. So that's the more straightforward of them because there's really only a couple of things that we're trying to detect. With smell or olfaction, we want to be able to detect airborne chemicals for a variety of reasons. And so there are far more chemicals that we can detect this way than we can with taste. Now, the old myth is that like we don't smell very well at all compared to other species, and probably that's not quite true. Probably we smell pretty decently based on current research. And the estimate is that we are actually capable of detecting about 1 trillion different scents, which is pretty cool. Now, it's not because we have 1 trillion different types of receptors, but it's because scents are often complex additions of multiple molecules. And so when you add all of that together, it ends up being about 1 trillion. So we have specialized epithelium inside of our nasal cavity, just in this small area that covers the superior nasal concha. And that's basically the top back of your nasal cavity, posterior and superior. So we have sensory neurons acting as the sensory receptors. These guys are bipolar neurons. And they are in there with a bunch of support cells that are basically little simple columnar epithelial cells. And then these guys also have stem cells. I think I've got a slide on that later. Just like the microvilli on the smell cells, on the taste cells are where the receptors are. These guys have essentially their dendrites sticking out like cilia. So we call them olfactory cilia, but really these are the dendrites of the neurons. And that's where the receptors are. And then the axons are non-myelinated, because honestly, they're not going that far. And they just pop through the little holes in the ethmoid bone at the base of the brain to get inside of the skull. Now, certainly species like dogs have much larger areas of olfactory epithelium, and they have proportionally larger olfactory bulbs. So see, we have like maybe 6 million receptors, and they have like 300 million, but we do smell pretty well, and they have actually tested it. So here are our bipolar first order neurons. So here's the dendrites, the little cilia sticking out on the surface of the olfactory epithelium. It's going to be covered in a little bit of nasal secretion, so mucus. And the support cells have cilia, and they are moving the mucus. And so anything that comes in the air and dissolves into that mucus can potentially bind to receptors if we have them. And then the axons go through some connective tissue, through holes in the cribriform plate of the ethmoid bone, and they synapse in the olfactory bulbs, which is what we identify as cranial nerve 1 or the olfactory nerve, but it is in fact the place where the cell bodies of the second order neurons live. So the chemicals that these sensory neurons are able to detect are called odorants. So an odorant is any chemical that can bind to a receptor of olfactory sensory neurons. And these guys actually get replaced about every 2 months by basal stem cells. And that's actually crazy because neurons normally live for years, if not your entire life. And yet these guys are getting replaced every few months. But that's good because if for some reason you lose your sense of smell because you accidentally catch too much of a whiff of ammonia or something and it like burns it out, they'll all get replaced and you should be able to smell again, which is a good thing. This is a close-up of that. So again, bipolar stems, sensory neurons, dendrite endings that are called cilia acting as the site of the receptors, and then pathway back to the brain. And this is where a lot of the mucus comes from, little glands. Oh, that's where I put that. Cool. Okay, so like I said, an odorant is a chemical detectable by an olfactory receptor. And we have about 400 genes devoted to making different types of receptor proteins for these cells. Now, our sensitivity to different odorants varies. So basically, the number of receptors we have for different chemicals is variable. If we have a large number of receptors, then we're going to be very sensitive to that smell and detect it at fairly low levels. But if we have just a few of those receptors, then it's going to take a very high concentration of a particular chemical for us to be able to detect it. The other weird thing is that a couple of things that we think we smell, we actually don't smell. It's just that they cause pain or they bind to our temperature receptors within our nasal cavity. So like ammonia and capsaicin is the thing in like peppers that is spicy. They will actually stimulate our pain nerve endings, and that's why the burning sensation happens, excuse me. And then methanol actually binds to our cold temperature receptors, and that's why it feels so nice and cool. It's not the only one, obviously. There are a couple of them, but that's kind of a big example of it. All right, so I kind of talked about this already as far as like, in order to be smelled, you need to be in the air, and then you need to be able to dissolve. And then the way that we end up generating electrical signals is that they are ligand-gated ion channels. And so then this generates graded potentials, and if enough of them depolarize the membrane to threshold, then we get action potentials, and we send a signal that says that we smelled something. As I said, the sensory neurons are the ones doing the detecting, and once they go up into the brainstem, into the cavity of the skull, they synapse with the olfactory bulbs. And so the olfactory bulbs have the bodies of the second order sensory neurons, and then the axons that leave from the olfactory bulbs form the olfactory tracts, which take information to the olfactory epithelium. There's also pathways to the frontal lobe of the cerebral cortex, as well as the hypothalamus and the limbic system. Now, every other sensory input into the cerebral cortex has to go via the thalamus. And the olfactory pathway is the only one that circumvents that. And it circumvents that because it's a very, very old part of the brain. And I think it was there before the thalamus. So we get more raw smell data than we get of anything else. It's the least processed information that we get to our cerebral cortex. So this is a little thing of the pathway, receptor cells, olfactory bulb. Amygdala is part of your limbic system. So we have a really strong tie between our emotions and smells because our limbic system was originally the pathway for smell. And then we'll also be going to the cerebral cortex, and then we can go to the thalamus and go to other places. All right, any questions about chemical senses? So next we're going to talk about hearing as well as the vestibular sense, which is equilibrium or balance. And we're going to do them together because the specialized organs are both in the inner ear. So we'll start with hearing because that uses the entire ear and we'll go through all the anatomy. And then we'll get to vestibular sense. So we're going to have to touch on a little bit of the physics of sound because otherwise it's hard to understand how we're able to detect sounds. So sounds are actually vibrations of molecules, and they are through whatever medium that sound is traveling. So vibration of air molecules when we're speaking or otherwise making noises in the air, but sound can also vibrate water molecules. It can travel through solids, of course. That's why we can hear through walls and stuff unless they're properly thick or insulated. Basically, as long as there are molecules to vibrate, we can have sound. So the only time there isn't sound is through vacuum, which is a lack of molecules present. So vibrations are actually alternating waves of pressure. So the pressure increases and then it decreases. So basically we're pushing the air molecules together and then they're spreading apart. And that's how it's literally vibrating. And when we say that it's a sound, it means that we can hear the vibrations. Now that does not mean that all vibrations of molecules through a particular medium are hearable to us, but it means that we define sounds as things we can hear. And it's not the only thing we define solely by our ability to detect it. So here's a little visualization of how the changing pressure of the vibrations changes the density of the molecules, and that gets passed on kind of like a bunch of dominoes falling over, and so then the sound continues onwards. So we define sound by two characteristics. The first one is called pitch. So pitch is the frequency of vibration. And what that means is how many high and low pressure cycles occur in a given period of time. The measurement for that is called hertz. So hertz are cycles per second, as in literally how many up and down waves of vibration we have per second. We can perceive sounds that occur within 20 to 20,000 hertz. And we're most sensitive to things that are kind of in the middle of that. So in the 1500 to 5000 hertz range, which happens to be the range of human speech. So if something is lower than 20 hertz or higher than 20,000 hertz, it's still causing vibrations in the air or the water or whatever, but we can't hear it. So some visuals of pitch measured in hertz. That is the wrong direction. And then a little visualization of, so basically the human hearing range is here. So sounds can occur below that. We call that infrasound, and some species can hear it, like elephants. And then if it's over 20,000 hertz, we call it ultrasound. And again, some species can hear that. In fact, a lot of species can hear at least part of the ultrasonic frequencies. But bats and like dolphins and stuff especially can hear very, very high-pitched things. So the other characteristic of sound is how loud it is. So loudness is a function of the energy of the sound waves. And basically it's their amplitude. So you can have things of the same pitch that have different levels of energy. And so this would be louder than this one because the differences in the up and down are greater. So we measure loudness in decibels, and decibels are interesting because 0 decibels is the threshold of human hearing. Literally the quietest thing that a human being can detect. And so again, it doesn't mean that there aren't vibrations of the air or water that occur below 0 decibels. It just means that we can't hear them ever. Normal conversation comes in at about 60 decibels, and pain and damage to our ears is going to be somewhere over 100. So usually 120 to 140 decibels. So visual representation of decibels from, this one's actually in percentage, but I don't know what the percentage is. So 0 is here, and this is 100%. This would be very, very loud, and this would be non-hearable. Now, when we combine frequency and intensity, which is the other term for loudness, what we get is a map of the human hearing range. And so the 1500 to 5000 hertz pitch that we can hear is also when we're best able to hear the lowest decibel noises. And then as we get towards the edges of our frequency range, the sounds have to be louder in order for us to detect it. And I really don't know what happens beyond this, to be honest, because that's very, very loud. Just another one showing you how humans compare as far as their ability to hear different pitches. And then this is kind of showing you different things at different decibels. And notice that distance is relevant here. So of course, the closer you are to the source of a noise, the louder it's going to be, and vice versa. All right, so that's sound, and we'll get back into how that's relevant to our detection of it after we go into the anatomy of the ear. So the ear is actually divided into 3 regions. They are anatomically as well as functionally distinct. So the outer ear is the portions that conduct sound to the eardrum. The middle ear conducts sound to the cochlea, and then the inner ear converts sound to electrical signals. So let's look at each one. So outer ear is the part we see as an ear all the way down to the eardrum. So this is conducting sound. The middle ear is in here. It conducts sound to the inner ear, and then the inner ear is right here. And the nerves run to the brain. So the external ear has a couple of different parts to it. The fleshy bit that we call an ear is referred to as an auricle or a pinna. Those are interchangeable words. And it's skin, elastic cartilage, and some adipose. So like your earlobe is adipose. And the reason why we have those different textures to it, so the whorls and recesses and stuff, is because they're supposed to direct the sound into the external auditory meatus or auditory canal. So that's literally the tunnel into your temporal bone that ends in your tympanic membrane. The other word for tympanic membrane is eardrum, and your eardrum is a physical separation between your external or outer ear and your middle ear. Its job, because it's basically like the head of a drum, its job, which is why we call it an eardrum, its job is to vibrate in response to sounds. That allows it to transmit sounds to your middle ear, which is actually located inside of your temporal bone in an air-filled area called the tympanic cavity. Now, the tympanic cavity stays filled with air because it's connected to your nasopharynx. So it's connected to your throat by 2 tubes. Well, there's a tube on the right and there's a tube on the left, by a tube called the auditory tube. It's also called the Gustation tube. And then its truly proper name is the pharyngotympanic tube because it connects the pharynx to the tympanic membrane cavity. It's usually closed on the pharyngeal end, but we can equalize the pressure in our middle ear when we change altitudes. Usually when we yawn or even when we chew, we're opening that tube and we're allowing air to move in or out to equalize that pressure. Sorry, saying yawning and makes me yawn. There are ear bones in here. Those are the major thing, and we'll talk about that in the next slide. And then the connections between the middle ear and the inner ear are by way of a little guy called the oval window. The oval window is basically the hole into the inner ear, and we'll look at that next. Now, in order to get the vibrations from the eardrum or tympanic membrane to the inner ear, we're going to use the ear bones. So the fancy term for ear bone is auditory ossicle. And we have 3 of them in each ear. So they were named for what they look like. So the one that's touching the tympanic membrane, they thought it looked like a mallet or a hammer, so they called it the malleus. The next one was nice and big and kind of triangular, so they called it an anvil or incus. And then the third one is very weird shape, and it actually looks like the end of a stirrup that sits on a saddle on a horse. So stapes is the Latin for stirrup. Now, this part of the stapes is connected to that oval window, and so the vibrations get transmitted all the way through these bones and then into the inner ear, which we'll talk about in a little bit. There are also 2 muscles in here. They're like the tiniest muscles in the human body, the stapedius and the tensor tympani. Stapedius is the one connected to the stapes. I believe it is in fact the smallest muscle in the body. And then the tensor tympani is connected to the malleus because it can change the tension of the tympanic membrane. So here's another view of that: tympanic membrane, malleus, incus, stapes. And here you get a better view of the stapes is touching the wall of the middle ear so that it is covering that oval window. And then the round window is also called the secondary tympanic membrane, and it has another little membrane, and it's sort of the other end of the connection to the inner ear. We'll talk about that a little bit later. Another view, just because. Oh, and I should point out here, you can see that there are little ligaments suspending these bones as well to keep them in place. So the inner ear is literally a little maze inside of the temporal bone. So we call it the bony labyrinth. And inside of the bony labyrinth is a soft tissue membrane lining the whole thing. So that part's called the membranous labyrinth. And there's 2 types of fluid. So there's a fluid called perilymph between the bony and membranous labyrinths. And that's basically just the watery portion of blood, to be honest. And then within the membranous labyrinth is the endolymph, which is a very high potassium fluid. So this is our little bony labyrinth. So you can see the stapes here, oval window is connected to the membranous labyrinth, which is represented in blue here, filled with endolymph. And we have 3 parts to this internal ear. So the vestibule is the central portion. It's part of our vestibular sense, and it's subdivided into the saccule and the utricle. The semicircular canals are also part of the vestibular sense, and there are 3 of them labeled by their approximate orientation, which we'll talk about a little bit later. So there's anterior, posterior, and lateral. And then the cochlea is the only part of the bony labyrinth for hearing. It looks like a snail shell, that's what cochlea means. And there are 3 fluid-filled chambers within the cochlea, which we'll address so we can see how we're actually detecting sounds. So when you think about this whole membranous labyrinth inside the osseous labyrinth thing, or bony labyrinth, so I want you to think about, what if you took the snail shell and you rolled it out? Okay, so now you just have a tube. I need to make the tube bigger, hold on. Tube only works if the tube is big enough. All right, so we're gonna roll out this tube. Now later we can roll it into, you know, the spiraling snail shell. shape. Okay, so that's our bone. And then we're gonna put this nice little soft tissue membrane down the middle, like sticking a smaller water balloon into a bigger water balloon. We're gonna put perilymph surrounding the soft tissue, so perilymph between the two labyrinths. And we're gonna fill the soft tissue with endolymph. And then inside this soft tissue structure, we're gonna put the special sensory organ for sound detection, okay? So that's going to divide this up into 3 chambers. So the scala vestibuli is the superior chamber containing perilymph. So it's got bone on one side and it's got soft tissue membrane on the other. The scala tympani is on the other end. It's the inferior chamber, and it also contains perilymph, and it's actually continuous because once we roll this sucker up, we'll see that one of them goes all the way around, we come here, and it just kind of doubles back as the other guy. In between them is the scala media. It's also called the cochlear duct because it is the soft tissue part, and it's the middle chamber. Now it is completely separated by those membranes, and it contains the receptors for hearing as well as endolymph. So the spiral organ is what we call that. And then there's endolymph here, perilymph, perilymph. So when we then spiral it up and cross-section it, what we end up with is being able to see a bunch of different windows into this organ. So again, vestibuli is superior, tympani is inferior, and the scala media with the spiral organ is in the middle, okay? We have different names for the membranous dividers. So vestibular membrane is here, and then the basilar membrane is here. The basilar is the more important one. You can see that the cochlear nerve comes up the center of the bony part, and so the sensory neurons kind of connect with this spiral organ from the medial aspect. So the spiral organ is this entire, it's basically a strip-shaped specialized sensory organ. It sits on the basilar membrane, and it's composed of a combination of support cells and cochlear hair cells. So just like the taste buds are these epithelial sensory cells and support cells, so this spiral organ is these specialized sensory cells, which are not neurons, as well as support cells. These cochlear hair cells are called that because they have these little like specialized microvilli that are on the top called stereocilia, and they look like hairs. And there's 2 kinds, there's the inner and the outer. Inner and outer refer to their location relative to the center bony mass of the cochlea and where these not axons, but where the neurons are connecting. So the inner hair cells are closer, and then the outer hair cells are over here. If we look at detail of this, so you can see here's a hair cell. So the stereocilia are embedded in this gelatinous structure called the tectorial membrane. And then the base of the cell is the dendrite endings synapsing with the hair cells. So this is where we're going to have neurotransmitter release and potentially send hearing-related signals back to the brain. So the entire ear is set up to transmit sound through the middle, or through the outer ear, through the middle ear, to the inner ear, where we're then transducing those sound waves into electrical signals to the brain. So we direct sounds in via the pinna, transmit them down this external acoustic meatus. We vibrate our tympanic membrane. The vibration gets concentrated and transmitted through these ossicles, and all of it ends up transmitting through this round window. And then we vibrate all the fluid inside of the cochlea. So the tympanic membrane is actually fairly easy to vibrate, and the auditory ossicles actually have to concentrate the vibration. So they kind of amplify them because they have to vibrate the perilymph on the other side of that oval window. And it's harder to vibrate fluid than air. If you've ever been to the beach, or even if you've been in like a wading pool or something and you tried to walk through a foot or two of water, you know how much harder it is to walk through water than to walk through air. And so vibrations are the same way. It's a lot harder to vibrate water than it is to vibrate air. So we have to amplify those vibrations as we transmit them into the liquid form of the inner ear. Now, the reason why it's like that is because it's easier to protect the inner ear from damage, because we can't necessarily vibrate the fluid so hard that it damages those delicate hair cells. So that's why it's like that. All right, so we transmit those vibrations. Now the perilymph starts to vibrate, and the perilymph is, of course, in the scala vestibuli, and so the scala vestibuli is vibrating, and the soft tissue membrane, vestibular membrane between the scala vestibuli and the scala media also starts to vibrate. And that's going to make the endolymph inside the scala media vibrate, and that's going to make the basilar membrane on the other side of the scala media also vibrate. So we're vibrating all of it, and that's going to result in vibrations of the spiral organ. Its other name is organ of Corti, which is why it says that here. So everything is vibrating. Now, where in the spiral organ that it's actually going to respond to the vibrations actually depends on the frequency or the pitch. So low frequency sounds travel all the way up the scala vestibuli, and they actually come around the helicotrema, and they keep vibrating the perilymph down the scala tympani. So then they're vibrating the basilar membrane from that side. And then the vibrations actually keep going, and the scala tympani actually ends in that round window. The round window, or secondary tympanic membrane, then bulges to release the pressure of this vibrating liquid. Higher frequency sounds are actually able to just cross into the scala media from the scala vestibuli, so they don't have to go all the way around. Now, the reason why this happens is because the basilar membrane has different stiffnesses at different areas. So the stiffest fibers are near the oval window at the beginning. The longer fibers are near the cochlear apex. All right, so longer, floppier fibers near the apex respond to lower frequencies. The shorter, stiffer fibers near the oval window respond to the higher frequencies, and everything else is somewhere in between. So this is how your brain is able to distinguish frequencies or pitch, because it knows where along the spiral organ it's getting each neuron is coming from. And so it knows, like, oh, this part right here is being stimulated. That means we're hearing a very high pitched sound at 20,000 hertz. It'd be really hard to hear because that's right at the edge of our hearing. This is, I think, a prettier way to demonstrate that. But it's all the same concept. Okay, so which part of the spiral organ is responding depends on the frequency of the original wave, but that frequency is preserved. So see how this says frequency of standing wave is the same as the sound wave? The frequency is preserved in its transmission all the way to the inner ear, and that's how we are having the appropriate frequency bend the right part of the basilar membrane. Sorry. Okay, so how does that vibration result in electrical impulses? Well, that's what we're going to talk about. So hair cells are mechanoreceptors, and the stereocilia are actually connected to mechanically gated ion channels. So when they move, they are opening and closing the ion channels. And I say opening and closing because whenever they're moving, they're going to be moving back and forth simultaneously because of the vibrations. So we're going to be opening and closing these mechanically gated channels. When we're opening them, the ions are going to flow in and depolarize. And then when we close them, we end up with hyperpolarization, and it goes back and forth and back and forth. Every time they open, the potassium flows in. That's why endolymph is so high in potassium, because we're actually using potassium as our depolarizing ion. And then you don't really need to know this part, but when we depolarize, we actually trigger voltage-gated calcium channels. That leads to neurotransmitter release across the synapse. And now we are stimulating our first-order sensory neuron. So our first-order sensory neuron gets stimulated every single time we have these guys open, and then every single time they sway closed again, the stimulation stops. So the nerve impulses from the cochlear nerve, which is carrying all of these guys, end up being the same as the frequency that we're hearing. Now, all of that information is pretty much just being carried by the inner hair cells. The outer hair cells, their job isn't actually to detect sounds and send that information back to the brain. Their job is to minutely adjust the basilar membrane using smooth muscles, or the closest equivalent to that, in order to let the basilar membrane detect very accurately which pitch it's hearing. So those are reflexive actions that actually change the stiffness of the basilar membrane and allow it to kind of focus on the pitch that's inputting itself. Now, the way that it gets back to the brain is we have the sensory neurons, this is what I get for reading. So our first order neurons come in, they synapse in the brainstem, and then we're going to cross over. Remember, everything crosses over, and we're going to go up. There's another synapse here, which allows some reflexive crossing over too, and then we go through the thalamus and we end up in the brain. Okay, so these synapses allow for reflexive actions with respect to hearing. So lets us do things like look at things when we hear them, which I mentioned, I think, last time, if I remember correctly, as well as other things. Okay, so that's hearing in like a teeny tiny nutshell. Obviously a lot more to it than that, and when we hit vision, it's going to be the same thing, because we just don't have time to cover this stuff in detail, and it's probably beyond what we should be covering in the course anyway. So before we get to vision, though, we're going to touch on equilibrium, this fifth special sense, as we like to call it, because when we learn about senses as kids, we tend to overlook it. We learn about all four of the other special senses, and then we're taught that our fifth sense is touch. But touch is a general sense. It doesn't have specialized organs. It's not focally located on our head. this sense is. So by using the other parts of the inner ear canal, we're able to monitor our head's location in space. So now we're going to the vestibule and the semicircular canals, and we're going to look at how they detect essentially gravity as well as movement. So the point of vestibular sense is to track both the position and the movement of our head and neck. Now, the vestibular sense doesn't work alone. It uses all of the proprioceptive inputs from the rest of the body, and it actually uses visual input as well. And all of this adds together to help our head stay in place, basically. So mostly we're guiding reflexes that allow us to either change or maintain our head position as needed. When it goes wrong, basically we get very, very dizzy, and it can be hard to keep our eyes focused on appropriate things. If you're curious about that, here are some links about vestibular disease, which is what we generally call it when part of the vestibular system isn't working properly. Now, there's two parts to the vestibular apparatus, or our vestibular system. And basically they line up with the two different parts of the inner ear that we haven't talked about yet. So the vestibule monitors what's called static equilibrium. Now, it's static not because it's just about when your head's staying still. It's static because it's basically monitoring the position and the movement of your head with respect to gravity. So linear acceleration, meaning movement just forward, as well as just the position of your head when it's still. And that's as opposed to dynamic equilibrium, which is where the semicircular canals monitor changes to head rotation. So this is movement straight forward or back or whatever, and then this is rotation. So basically when you're, turning your head side to side, or when you're, head banging at a rock concert, something like that. So this isn't really about gravity, this is about our moving our heads, if that makes sense. So we'll start with static equilibrium, and then we'll hit dynamic. So the vestibule is divided into two subparts, the saccule and the utricle. And each part has a sensory receptor called the macula. Macula just means spot. So they each have one, and they work the same way. So we're just going to talk about it once, and that's how it works for both. Very, very similar setup to the spiral organ, but instead of there being a whole strip of it, now there's a single little spot of it. So it's a little patch of hair cells, and they also have stereocilia. And they also have a little membrane that the stereocilia are embedded in, but this time it's called the otolithic membrane, because on top of the jelly-like part are a bunch of calcium carbonate crystals. So basically these little rocks. So here's what that looks like. So hair cells and support cells, connections to our synapses with our neurons, stereocilia embedded in the otolithic membrane, and then our little otoliths on top, okay? So we're gonna have one of these in the saccule and one of these, one of these in the utricle. So it works basically when your head is tilting forward or tilting backwards. These act as a weight that moves the jelly-like membrane in the same direction as your head. So if you tilt your head forward, gravity is going to pull everything forward and bend your stereocilia in that same direction. If you're moving backwards, it does the same thing in the opposite direction. Oops. And we have the same thing happen. They're mechanically gated ion channels. If we go one way, we open the channels and we let potassium in, and that's going to increase neurotransmitter release here, and we get a signal. And then when we go the other way, we hyperpolarize, and we have no signals. And your brain knows which way is which. So in a teeny tiny nutshell, static equilibrium, okay? And so this is stuff like, not only is it, we know when we tilt our head forward, but it's like when you're in a car and you stop suddenly, that's going to register as well. So then the dynamic equilibrium, we have a sensor in each semicircular canal or duct, and they're named with by their approximate orientations to the 3 planes of space. So we have an anterior and a posterior, and then we have a lateral semicircular duct. And so that gives us a 3-dimensional rotational space to monitor. And each one has its own sensory receptor. So this time it's called the crista ampullaris. And so again, we have hair cells and support cells, and we have a little gel thing, but now it's in like a weird little troll cap. And they're each sitting at the base of each canal. And then of course we have synapses with neurons here. So looks like that. And you can see they're sitting here. That should be it. And so the way it works is, if you're rotating your head, the endolymph within these canals starts to move. And it's going to move in the opposite direction of your head. So if you're moving your head one way, the fluid moves the other way, and then it bends that little troll doll gel cap of a cupula, and that's what bends the stereocilia and opens or closes the mechanically gated ion channels. So the more you rotate, the stronger that signal gets. And if you rotate a bunch and then you stop, that fluid is going to keep flowing and keep bending your sensor. And so that's going to make it feel like you're spinning even after you stopped. And so that's why, like, when you're a kid and you do the whole spinny, 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 and then fall back and just watch the world rotate, that's why that happens, because your poor little sensors think you're still spinning for a little while until the fluid finally slows down and stops. So all of this stuff has to go to all of the areas that deal with coordination and balance and stuff. And so basically, we're going to head off to the cerebellum from the pons, and we're gonna have various reflexes in the brainstem concerning turning our head, and then we're gonna go cross over via the thalamus and go up to the cortex. But this cerebellar pathway is pretty important because it all ties into the same types of things that the cerebellum and the basal nuclei are dealing with. All right, any questions before we get to eyes? Okay, eyeballs. So eye anatomy and eye physiology is probably the most complex. So there's probably more anatomy to the eye. So we're going to go through it a little bit and just see that all of the eye stuff has functions. So when we talk about the eye, we really are talking about the orbit and the orbital region. So the orbit is the actual bony socket that the eye sits in, and the region is all of the structures related to protecting the eye and serving its functions. And so that includes your eyebrows, eyelids, and eyelashes, as well as some other structures like your conjunctiva, which is the mucous membrane that covers the outer surface of your eye, the lacrimal apparatus, which is your tear gland and tear ducts, and the extraocular eye muscles, which we talked about when we got to cranial nerves, that actually move your eyeballs. So eyelids and lashes, of course, protect the eye. Here's your lacrimal gland, which makes tears, releases the tears here. The tears go this way. We'll talk about it more later. And then they drain out like this. This is the medial caruncle. It's just a little fleshy mass, but there's a little gland in there too. And then we have all sorts of names for all of this stuff, but we don't really have time to go into all of it. So remember that we have 6 extraocular eye muscles, 4 rectus muscles, and 2 oblique muscles. And this shows you kind of what happens to turn the eye when each one contracts. And we're not going to go into them a ton because we basically covered them with cranial nerves. Where tear production, these are the different structures involved. So the lacrimal gland is the one that makes the majority of the tears. It gets secreted by little ducts, and then it travels across your eye to the medial aspect and drains out via little holes called puncta by teeny tiny tubes called the canaliculi. And then they collect in the lacrimal sac, and they drain into your nasal cavity by the nasal lacrimal ducts. This drainage is why when you cry, your nose runs more, because you're literally crying into your nose. So the tear film has a variety of functions. Most importantly, it provides nourishment to the surface of the eye. And then the rest of it is basically helping with that. So the watery part of your tears nourishes and protects your eye, especially your cornea, which is this clear part here. And then there's a mucousy layer that helps keep the tears stuck to the surface of the eye. And then there's oily parts which prevent the watery part from evaporating. And the mucousy part and the oily part are why when your tears aren't always draining properly, like overnight, you get those crusts in the corners of your eyes. This one's showing you the flow of tears out into your nasal cavity. So when we look at the eyeball itself, we actually break the anatomy down as it's a 3-layered hollow ball. So there's 3 layers to the wall of the eyeball, and we refer to them as tunics. We also look at the anatomy as far as which parts are see-through that let the light in so we can detect it. And there's a couple of parts to that also are parts of these tunics. And then we also look at which parts are nervous tissue. So the neural components are the only nervous tissue, that's the retina and the optic nerve. But all of it is going to be part of one of these 3 layers or tunics of the wall of the eyeball, or it's the fluid inside. So the tunica fibrosa is the outermost layer. It's mostly thick, dense connective tissue, so it's providing protection and structure to the eye. That's the sclera, or the white of your eye. And then the see-through part at the front is the cornea, and that's the part that your contacts go on if you have contacts. Now, just inside of that is the vascular layer. So the tunica vasculosa, it's also called the uvea, and this is where the majority of the blood vessels are inside your eye. And it's got a couple of different structures. So the choroid is the majority of it. The ciliary body is the part that anchors the lens. The iris is, of course, the part of the eye that we can see that has color. And the middle of the iris is hollow or empty, something like that. And that's what we call the pupil. So the pupil is not a structure, it's a space inside the iris. The innermost layer is called the tunica interna, so the most internal layer, and it's all nervous tissue. So it's just the retina and the optic nerve, and we'll look at that later. So if we look at an eye diagram here, we'll see the sclera protects the whole outside of the eye, And then the cornea is just where it needs to be see-through to let the light in. So that's the fibrous layer. And then the choroid forms the majority of the vascular layer. And then the ciliary body is here, suspending the lens. And then the iris is up here. And then the internal layer is the retina, which is this yellow stuff here. It only goes up to here, okay? And then it is just attached to the ciliary body by this structure called the ora serrata. It just means the serrated opening, orifice, doesn't matter. And then back here, the retina flows into the optic nerve, which we can see as a disc inside the eye. So the optical components have to be transparent to let the light in, but they also focus the images so that they're clear and detailed when they hit the retina. So the components of that are optical are the cornea, which is part of that tunica fibrosa, the aqueous humor, which is the fluid at the front of the eye, the lens, which is that big clear thing that I pointed out, and then the vitreous body, which is the fluidy stuff in the back of the eye. The iris has two smooth muscles that control the amount of light in the eye. So when we talked about that pupillary light response, these are the muscles that are dealing with that. So these are under autonomic control. One of them is a circular muscle called the pupillary constrictor or sphincter, and the other one is called the pupillary dilator. So because these are smooth muscle, they are involuntary and reflexive. So the dilator is all of these straight, smooth muscle bands between the edge of the iris and the edge of the pupil. When the constrictor functions, it's of course going to constrict the eye, and that's parasympathetic. And when the dilator contracts, it of course dilates the eye, and you can see it just kind of pulls everything open, and that's under sympathetic control. Oh my God, sorry, I'm itchy. Okay, now, as you saw in the diagram, there's internal spaces to the eye as well. So we divide up the eye into two segments. Basically, there's a front or anterior segment called the aqueous chamber, and it is further subdivided into an anterior and posterior chamber. And then the lens is our divider between the anterior and posterior segments. And then everything behind the lens is the posterior segment or vitreous chamber. So the aqueous chamber is filled with aqueous fluid or humor. And then the vitreous chamber is filled with the vitreous body or humor. Honestly, this one is mostly a gelatinous thing, and this one is an actual fluid. So the posterior segment or vitreous is all of this back here. Basically everywhere there's retina is posterior. And then here's the lens. So the lens and the ciliary body here are dividing us up between posterior and anterior. And then the iris divides the anterior segment into an anterior chamber and a posterior chamber, which is pretty much just the space between the lens and the iris. Now, hold on one second. Okay, yeah. This is just kind of a close-up of all that because I found a cool picture and I liked it. But the aqueous is constantly being formed. It's not that dissimilar to CSF or cerebrospinal fluid. So the ciliary body makes the aqueous and it flows out across the top of the lens, and then it actually comes through the pupil into the anterior chamber and then it drains out the edges of the cornea. Oh my God, it's gonna, hold on. Don't mind me. Okay, so yeah, so it's constantly being produced. And it's constantly being drained back into the veins. The vitreous, on the other hand, is just kind of a gel blob, which I think they show well here. And that just kind of maintains constant pressure. It kind of holds the lens up there. These little ligaments hold the lens in place, but it kind of helps it stay up there, and it holds the retina to the whole back of the eye as well. Oh man. Okay, so the retina is technically the only part of the brain that can be viewed from outside of the skull, which is a little bit crazy in my opinion. And as I mentioned, it's only actually attached at the ora serrata, and then actually at the optic disc where it is becoming the optic nerve and exiting the eye. Now, the most important part of the retina is a spot called the macula lutea, means the yellow spot, which contains the fovea centralis, which has the highest density of the photoreceptor cells to detect vision. This is where we're actually able to see very high detail, high resolution stuff. The rest of our retina isn't actually nearly as important for actually seeing stuff. And the photoreceptor cells, of course, are the ones that are detecting light and converting them into electrical signals, which we'll talk about more in a second. And then that optic disc is the part of the optic nerve that we can see. And again, it's the only nerve that can be viewed from outside of the body, which actually makes examination of the inside of the eye a fairly important exam for purposes of detecting certain diseases. So this is actually what it looks like when you have the inside of your eye examined. So we call it a fundic exam. This is also called a fundus. And this pale disc here is the optic disc. So that's the optic nerve, cranial nerve 2. And then the macula lutea is right here. So this is where we actually are trying to focus all the light that's coming in, because this is what we want to be very, very, we want this to detect everything. And then all these little branchy guys are actually blood vessels. So there's a central artery and vein that runs down the middle of the optic nerve, and they actually come out and supply the retina with nutrition. All right, so the retina is where light detection happens, right? And that's because it has photoreceptors. Photoreceptors are modified neurons. So they're highly specialized. They have a bunch of visual pigments that is able to respond to different wavelengths of light in the part of them called the outer segment, which kind of looks like this. And then they have an inner segment, which is where the cell body is. And then they have a little axon and a synapse, where they synapse with the first true neurons called the bipolar cells. There's 2 different types of photoreceptors based on the shape of their outer segments. Rods have rod-shaped segments, cones have they're shaped like cones. Okay. Now, rods only have 1 type of visual pigment, so there's only 1 type of rod total. And then cones have 3, so there's 3 types of cones. And when we say visual pigment, we're talking about molecules made up of a combination of a form of vitamin A called retinal, as well as a type of molecule called opsin. And this combination is able to respond to visible light. Oh my God, probably should write on these things. Okay, so you can see we've got retina and rods and cones. Yeah, it's fine. Okay, so before we really talk about how rods and cones let us see, we need to touch a little bit on what we're seeing. So just like we talked about what what sound is, we need to talk a little bit about what light is and what we're actually detecting. So light is actually electromagnetic radiation, you know, like what the sun gives off. We can only detect it within a very, very narrow range of the total range of electromagnetic radiation. And of course, for most of ever, sunlight was the primary source of energy as well as light for the planet. And now, of course, we can turn on light bulbs and stuff. So we're not always dependent on the sun for light, but we are definitely still dependent on it for a lot of energy. The form that light is emitted from the sun in is photons. And when it comes out, it's coming out as basically, waves, as waves. So just like sound has wavelengths and frequencies, so does electromagnetic radiation. And the wavelength that we can detect is the range between 400 and 700 nanometers. Beyond that, there is still electromagnetic radiation, but we don't call it visible light because we can't see it. Just like there's sound above and below what we can hear. So the whole electromagnetic radiation spectrum runs all the way here, and we see this teeny tiny sliver. Stuff that is at a lower wavelength is ultraviolet light, X-rays, and gamma rays, and stuff that's above that wavelength is infrared and radio waves. Okay, so we have electromagnetic radiation, and we have photoreceptors with pigments that are set up to detect it. So the photoreceptors actually, when they're not exposed to light, are constantly firing signals across their synapse with these bipolar cells, inhibiting the bipolar cells from firing. It's a very, very weird setup. So once light of the right wavelength stimulates the photoreceptor cells, the photopigment molecules change their configuration. It changes their shape. And that shape changes the electrical activity of the photoreceptor cells, so they stop firing their inhibitory signals. And because they're no longer inhibiting the bipolar cells are able to fire a signal. That signal is going to make its way into the brain, and now the brain knows, like, oh, our eye is seeing this wavelength because we're hearing from the bipolar cell. And the bipolar cell is actually the first order sensory neuron of the visual pathway. So here is a photoreceptor cell chock full of pigment in its outer segment. And this is where it synapses with a ganglion. And under ordinary circumstances, it's, like I said, constantly releasing neurotransmitter, which inhibits the bipolar cells. And you don't need to know this, but it's because they have open sodium channels, and so they're actually able to depolarize and release. When you expose it to the appropriate wavelength of light, though, the change in the pigment closes the channels and hyperpolarizes these guys, and now there's no neurotransmitter release. So now the bipolar cell is able to send signals. All right, so why do we have different types of photoreceptor cells and different types of pigments? Okay, so the rods are able to see black and white because they only have one type of pigment. So they're either on or off. They can see something or they don't. And they work really well in dim light. They don't work in bright light. The cones, because there's 3 different types of pigment and therefore 3 different wavelength spectrums that can stimulate them, see color vision, but they require bright light. Dim light is too dark for them and they can't work in those conditions. So we have 3 different types of cones based on the wavelengths that primarily stimulate them: short, medium, and long wavelength. where this is relative to that 400 to 700 nanometer range. So here's the total range of human vision, 400 to 700. Our rods see this. So light that is most likely to stimulate the photoreceptors is right around 500 nanometers, but light from like 420 to almost 600 can still stimulate. So anything in this range, we're going to register as a brighter spot in our black and white dim light vision. When we look at our three types of cones, we see that our short wavelength cones see best at around, blue, but there is a range here, and that range overlaps with our medium, which see best as green, and we like to think about these as red, but they're really more yellow, if that makes sense, as far as what they maximally respond to. And so there's a lot of overlap here. And so the reason why we have 3 cones, but we see more than 3 colors, is because the percentage of our cones stimulated varies across this entire range. And so you can see that it's showing you at different wavelengths, different percentages of each type of cone are stimulated, and our brain knows that that means it's a different color. So for example, red is the only color where none of the mediums and only long wavelengths are stimulated, and that's how we know it's red. But as more and more of our mediums are stimulated, we go to different shades of orange, and then we get yellow, and then we get green. And once we start stimulating our short wavelengthers, now we're introducing a bit of blue in there. So we're getting the aquamarine blue greeny stuff, and we get blue, and then ultimately we get purple or violet. Okay, so that gives us the whole range. Now the reason why we have 2 different systems, essentially we have 2 different types of eyes in the same eyes, is because of that difference in dim light versus bright light. So rods are very sensitive to light in general. That's why they work really great in dim light. It doesn't take a lot to activate them. But they don't work in bright light. They literally get blinded, okay? They're blinded in bright light. So they're not great in high light conditions, but in low light conditions, they give us night vision. And they work pretty well in our peripherals too. Now, we don't have very good detail with our rods because 100 rods typically synapse on a single ganglion cell. Now, the ganglion cells are the next step. So we have the photoreceptors synapsing with our bipolar cells, and then the next step is our ganglion cells, and they send out axons to become our optic nerve. Oops. So 100 rods per ganglion cell is like having really big blocky pixels when you're trying to make a picture. You don't get a lot of detail when your pixels are very large. If we contrast that with cones, so cones don't work in low light conditions, they only work in bright light. So, not great when we're trying to see in the dark or in really low light conditions, but awesome when we have full daylight out. They're faster to react than rods. And we only have 1 cone synapsing per ganglion cell. So now instead of these giant pixels, we have a bunch of teeny tiny ones. And so the detail level that we can see with cones is way, way higher. So we either have high detail, high color, bright light vision, or we have low detail, black and white night vision. But we like having both, right? It's very, very important to be able to see in the dark. And yet it's very useful to have this high definition vision in high light conditions. And so that's why we have both, because they both have pros and cons, and we wouldn't do nearly as well as a species if we only had one or the other. Now, the consequence of this is that when we're switching lighting conditions, when we're going from very bright to very dark or vice versa, we have to adapt because we're switching from one vision system to the other. So light adaptation is when we adjust our vision going from a dark area to a brighter area, which means that we're going from using our rods to using our cones. Now the cones have been inactivated, and we walk out with our dilated pupils and they rapidly constrict because of our PLR responses. And our poor little rods get blinded, but our cones have just been resting and waiting. And so it only takes about 5 to 10 minutes to achieve full visual acuity as we go from dark to light. Now it's a little bit different when we go from bright areas to dark areas. So dark adaptation is when we do that. And so now we're going from cones to rods. So of course, we're still able to rapidly adjust our pupil size. So our PLR gets turned off essentially, and we get a rapid pupillary dilation to bring in more light. Our poor cones aren't getting enough light now, so they can't function anymore. But our rods have been blinded in this bright light conditions, and so they actually have to reset themselves because they've been just overwhelmed by the light coming in. So they have to reset and then slowly start working again, taking in these light conditions. So it takes 20 to 30 minutes to achieve full night vision acuity because the poor rods are just starting with like half a deck, you know? Like they have all the cards stacked against them, and the cones typically have all the cards going for them. All right, so that's the other thing about it, is switching back and forth literally means switching vision systems. All right, so the last thing we're going to talk about is all those optic components that are actually bringing the light to the retina. So we talked about the anatomy, we talked about what happens at the level of the retina, but now we're going to talk about the pathway of light and how we're actually focusing light on that teeny tiny fovea centralis to achieve that visual acuity. So we have one more physics thing, and it's light refraction. So light refraction is the bending of light when it's passing through regions of changing densities. So the best example of that is that light is going to get bent when it's traveling, or differently when it's traveling through air versus water. So here we have a straw in a glass that's mostly filled with water. Now technically there's a little bit of refraction as it goes through the glass as well. So we just get this little blip here on the edge of the glass where it's particularly thick. But of course you can really see the refraction difference when you see where the straw is entering the water from the point of view of the air versus the point of view of the water. Now we know that this straw is continuous, and yet it looks like it takes a very, very drastic bend, right? And that bend is a consequence of the light bending, not the straw. So that's light refraction. So the optical components of the eye are going to refract light because the light is traveling through air, and then it has to travel through see-through tissue as well as some fluid. The two parts that are actually refracting are the lens and the cornea because they are curved surfaces. So a lens is any curved surface that can refract light. When a lens is convex, as our lenses are, they're curved outwards on both sides. So they're the thickest in the center. And when light comes through, it gets bent inwards. And that leads to the light converging on a single point called the focal point. The thicker the lens, the closer the focal point is to the lens itself. So if I have a thinner lens than that, then it doesn't bend the light as much, and the focal point is farther out. And then if I had a really, really thick lens, then it would be really close. Good enough. The other thing that happens when we refract light is that the images get flipped. So you can see up here, So like we've got a yellow corner here, and it goes through this lens and it gets refracted so that instead of being in the bottom closer to us corner, now it's in the farther away upper corner. All right, so things get flipped upside down and backwards. We call that image inversion. And it's what happens when you go through a lens that's bent on both sides. So the cornea refracts the light a lot. The cornea, though, is only bent one way, so it's convex outwards. So it refracts, but it doesn't invert the image. But then when we go through the lens, because the lens is convex both ways, now we're converging it a little bit more, but we're also inverting it. Now the cornea does the majority of the refraction, but the lens does the fine tuning, and the lens is able to change its shape, and that means it can adjust how far out it's focusing, and the cornea can't do that. Now the reason why, even though everything entering our brain or entering the back of our retina is upside down and backwards, but what we see as normal is because our brain makes the adjustment. So our occipital lobe, receiving all our visual information, is actually going to flip the image back based on experience. Oh no. All right, so when we look at how we change focus, what we see is that when we want to look at things close up, we have what's called a near response to vision. So we actually have to work harder to see things close up than we do to see things far away. Generally speaking, on average, people are able to see things that are 20 feet or farther away without having to make adjustments for close range vision. Once you get closer than that, you have to actually start doing more work with your eyes. I mean, you're not aware of it, but you are, in order to see closer up than that. So there's three things that we do when we're looking at things closer than 20 feet. We do something called lens accommodation, which I'll talk about in a second. We also have pupillary meiosis, which is literally constriction of your pupils as you look at something closer up versus farther away, because literally you're looking at a smaller field and you don't need as much light to come in. The third thing that we do is eye convergence. So eye convergence is how our eyes actually start turning inwards because we're looking at something that's closer to us. And eye convergence is why we can cross our eyes if we focus on an object and move it towards our nose. It's just crossing your eyes is just a consequence of the ability to converge your eyes. So lens accommodation is how we change the shape of the lens to change the focal point. And the reason why we need to change the focal point is because once we're close enough to stuff, we need to converge the image more in order to get it to actually focus on the back of our retina. So the way we do this is because the ciliary body contains a circular smooth muscle, just like the pupil does. And then the lens is suspended between that muscle, or the lens is suspended within that ring of muscle by these little suspensory ligaments that are also called ciliary zonule fibers. So when those fibers are taut, the lens is being pulled and it's fairly flat. And that happens when the ciliary muscle is relaxed, because remember, circular muscles, when they contract, they get closer, right? They make the hole inside of them smaller. And when they relax, then they're wider open. So when the ciliary muscle is relaxed, that lens is taut and it's a little bit thinner. And then when we want to increase the convergence, because we're looking at something closer up, we actually constrict the ciliary muscle. That actually brings it closer together. The zonular fiber tension decreases, and the lens naturally bounces into a thicker shape. Now we're going to have more convergence, right? So what that would do is focus it on an area closer to it, but what it does in this case is because we're seeing something that's closer and therefore the light is coming more, it just focuses a closer image. If that makes sense. Please tell me I have a picture. I do not. Awesome. Okay, well, try that again then. Okay, so basically when you're at least 20 feet away, everything's coming in and you naturally focus it on the back of your eye. Okay, but when things are closer, if we just stuck with the normal way that we were doing things, because it's now a bigger thing, it's coming in and, oh no, I did that again. And, oh boy. And it would accidentally focus too far. So we need to then move the focal point by thickening the lens to get it to actually focus on the back of the eye again, okay? So that's called lens accommodation. And it's the major player in how we see things up close. It's also the thing that starts to fail as we get older. So as we get older, the elasticity of the lens fails, and that's why we start getting very nearsighted, or why we need reading glasses and stuff, right? Like when you get older, you end up like holding things really far away from yourself because you cannot make your eyes focus as close anymore because your lens doesn't bounce as thick as it used to. You can't do this lens accommodation thing anymore. The other thing we can do, because we have two eyes that are able to look at the same thing at the same time, is that we have depth perception via something called stereoscopic vision. So stereoscopic vision is our ability to tell how far away something is, which is another way of saying depth perception, or it's basically the same thing. And you have to have two eyes that are looking at the same thing, but because your eyes are slightly apart from each other, they're looking at the same thing with a slightly different image. And we know that because we can hold something in front of our eyes and we can close one and we can close the other and we can see that it's not quite the same image. But when we have both of our eyes open, it looks like we're looking at one thing. So because your eyes do that, and then our brain uses something called a fixation point to judge relative distances based on focus. As in, if I'm looking at two objects and I'm focusing on one of them, everything that I can also focus on at the same time must be the same distance away because of how focus works. If it's blurry, then it's either closer or farther away than the thing that I'm looking at. Okay, so that's what fixation point means. And so that's kind of represented by this diagram where we're looking at these two things, and if they're focusing, they're both nice and focused, that's because they're the same distance away. Whereas if we're looking at two things that are not the same distance away, they don't focus the same way, is basically how that works. So the last thing to look about, the last thing to see with vision is the idea of the visual field. So the visual field is everything we can see with both of our eyes. And what we see is that the left visual field is here and the right visual field is over here. But stuff from the right visual field hits both eyes, and stuff from the left visual field also hits both eyes. Okay? And obviously there's some overlap in there, in the middle. That's where we have stereoscopic vision. So because we like everything from one side of the body to be processed by the other side of the brain and vice versa, we do the same thing with our visual input. So everything from the right visual field, we want to get processed by the left occipital lobe, the left visual cortex. And so the what's coming into this eye stays on the left side because it's already there. And then this crosses over at the optic chiasm, which is the little X right behind the optic nerves, so that it too ends up on the left side of the brain. And the same thing happens to the right, so that the left visual field, all that information ends up on the right side of the brain. Okay? And so this is also partly how we have really good stereoscopic vision, how much crossover, it does vary by species. And so we have enough to have really good depth perception, 3D vision, and stereoscopic vision. Just like with hearing, excuse me, there are some synapses in the brainstem as well. And those synapses are why we have reflexes related to our eyes. So again, in a nutshell, because we don't have time to go into a ton of detail, that is special senses. And that's a summary about eyeballs that I left in because it was fun. Any questions? Oh, well, that sucks. Yeah, I'll put it up tonight. So the only controlling factor as far as how long